It's my great pleasure to welcome Manu Prakash here for our Institute Colloquium online. And Manu is a professor for bioengineering at Stanford University. He studied, I think, informatics in India at the Institute of Technology, right? And before he moved to Boston, to MIT, to do a PhD in physics. And he was then a fellow at Harvard University, but already kind of on the move towards Stanford to start his own lab, where he has been, I think, for 10 years, 11 years or so. And I met, before he became famous, I met him because his wife was actually postdoc in the lab where I postdoc, so we had dinner once. And then only later I saw what kind of amazing science he does. And what I really like about his work is that I think Manu can just look at things and then he finds something extremely interesting and makes beautiful papers out of them with really interesting biology and physics at the same time. And it's really remarkable. So every time when I hear a talk from him, so I think last time was at ACB in Washington, I'm just blown away how pretty everything is and how nice. And he's not only a fantastic scientist, but he's also interested in democratizing science and make science accessible to everyone. So he's I think most famous outside of science for his invention of the foldscope, which is a cheap microscope, which I think opens up the beauty of small organisms to everyone. So I think that's really great. And he also made a paperfuge, a centrifuge out of paper for doing science experiments at home. So yeah, I think he's a really important person in biology, soft matter, and self-assembly, and also in this frugal science, is how he calls it. And so today he's going to talk about how protists, I think, drop, or so how gravity shapes life on Earth. And I'm really looking forward to his talk. So because I'm not sure who of you have attended this kind of format already. So the way we do it right now for the questions is that you have this Q&A button on the bottom of your screen, and you can just click that, type in your questions. And at the end of Manu's talk, I will read out those questions, and he's going to answer them. And then for all postdocs and PhD students, so what I'm planning to organize, and I hope it's going to work out, is a Q&A session with Manu at some time during the week. So I hope we find a time where I just organize another Zoom meeting, and then postdocs and students can maybe ask questions to him. So he volunteered for that, which I think is really great. Okay, so now I let him start. Thank you, Manu, and I'm looking forward.
Thank you, Martin, so much. This is a strange format because we're all probably taking this from the luxury of our homes. I just want to thank Martin and everybody for the chance to be able to still share science. And I think you've all seen what's unfolding around the world. And I think amongst all the news that we've seen throughout, I think for me personally, just the resilience of the scientific community, how people have actually come together, and the act of openly sharing science and solutions in this pandemic has been a remarkable thing to watch. And I hope that, beyond this pandemic, we can look forward to much more interactions across the world. This being one example. And I think in the very end of the talk, if there is time, I can briefly talk about what we've been doing in the context of COVID. But I wanted to take this hour to tell you about something that we've been working on the last 3 or 4 years, but it's starting to get to a point where we're very excited about starting to take some of these, both the technologies and these ideas out to the field. And that's what I'll talk about today. It's called a sinking feeling. And we'll really think a lot about gravity in this talk. And I don't know how many of you have been to the ocean or are sailors or might have peeked curiously in what's out in the ocean from a context of cell biology and just life in the ocean. And I think if you're going to take a perspective for today's talk, just look at this painting on the right. And I want you to imagine just like if you were walking in canyons in Arizona, imagine if you could do that for the ocean. So just keep the analogy of canyons in your mind and oceans. We will talk a lot about depth in the ocean and how do you study and understand how important depth is in this perspective. So let me know if you can see my slides move and hear me, right? Right, Martin? Okay, yeah. So I think I want to start with this this quote. This is from one of my favorite authors, Rachel Carson, and she's most recognized for her work in the Silent Spring and the environmental crisis, but she was a marine biologist and a marine author first. And what she wrote, and I'll just read a small portion of this, it is, "The ocean is a place of paradoxes. It's the home of the great white shark, the 2,000 pound killer of the seas, and the 100-foot blue whale." And this is the sets of images that come into our mind when we think of the ocean. But it's also the home to the small. And if you might scoop through with your two hands, there are stars, as many organisms as there are stars in the Milky Way. And she goes on and on to describe plankton, which is the most abundant life form in the ocean. And sort of in the very end, she states something along the lines of drifting endlessly midway between the sea of air above and the depths of the abyss below. These strange creatures and the marine inflorescence that sustains them are now called plankton, the wanderers. And this word plankton by itself just implies things that have no control, that are just there and are wandering at the mercy of the ocean. And we'll try to question that premise a little bit, but also just try to understand why it's important to be thinking about these things and what can we learn from a context of cell biology in this perspective. I want to start with a picture of the famous Dreyers rhinoceros from the 1515. This is a painting, a very famous painting of a rhinoceros made by an artist that had never seen a rhinoceros. And this was early time. This was primarily drawn by descriptions of an Indian artist that had sent some descriptions to London of someone who had actually seen a rhinoceros before. And this is a very common case in biology. We have to construct complete abstract notions out of incomplete information. And so if some of you might have seen a rhinoceros in a zoo and it might appear as a very docile animal, and you might see a rhinoceros in the wild and you might have a completely different picture. But in science, we have to be thinking about, can we observe the life that we care so deeply about studying in its ecological context to truly understand why it was built and designed the way it was? So I think in this talk, you will hear quite a lot about different pieces and how to put the pieces together, from being able to observe cellular scale behavior in the context of ecology. And I think one of the arguments that I'm trying to make here, and something that we've been working hard on, is without that ecological context, it's almost impossible to understand these remarkable cells. So these are the cells that I'm talking about. This is the very famous Haeckel's drawing of Radiolara. These are some of the most famous cell biology paintings that are out there. But ironically, if I ask you a very simple question, it's the same painting. I've flipped it upside down. Which one is correct? Do these cells have orientation? So in the context of the planet, should the Radiolara be drawn upside down or downside up? And I think by the end of the talk, I'll give you the answer to this. It sounds like a very simple question, but actually fundamentally, if you think about these cells are breaking polarity, these cells are constructing these artifacts, but without ever observing them in an ecological context, I cannot differentiate and decide what is correct. And you wouldn't be happy if I had made a rhinoceros upside down, because you would not be able to infer that legs are used for walking, And so, this is just one teaser. And I want to, in your back of your mind, be thinking about this question. And how would you address this question to begin with? Okay, so I'm going to start with a visual. And the movies are a little bit flaky. They're a little bit jittery here, but we'll have to just, I guess, bear with it. What you're watching is a marine larva. This is Pteria miniata. It's a starfish called bat star. Any of you who have gone tide pooling can often see this starfish. You can hold it. This is the larval form. But what's remarkable about the video that you're watching is you are traveling with this starfish in its frame of reference. And so for the very first time, we have been able to image cellular scale details of organisms, microscopic organisms, as if they are completely free to move. And what that allows us to do is truly study behavior of an organism in, say, a vertical column at a cellular resolution. And so, if you're thinking about in this jittery movie, what's actually happening is, I'm going to see if I can play this again. It's really slowed down, but what you can see is it's climbing up and it's rotating like a helix. And much of that movement is essentially associated with tiny, tiny, tiny cilia. We study these ciliary bands in this system. And one of the perspectives of this would be, so now here is an example of another organism, something that might be more familiar to a few of you. This is Volvox now climbing. And you start realizing that it's not just climbing. It's essentially, it climbs, it stalls, of course, it's spinning across. And of course, all of this behavior is in the context of light, chemistry, the chemical patterns that are in this water column, of course, the circadian rhythms associated with this. And one of the things that I'll tell you about first briefly is the set of tools that we have invented to allow us to be able to image organisms traveling in the vertical axis, infinite distances, if you will. And then secondly, I'll tell you about the sets of questions we can ask and understand in the context of life in the ocean with that tool. So before I start, this is some of the team members involved in the lab, with a graduate student, Deepak, another graduate student, Hong Chuan, postdoc, Adam, and then another intern, Delphine. And now another person that's not pictured in this is Ethan Lee. And one of the tools that we invented to study vertical migrations in the ocean, we call it the gravity machine. And it'll become apparent pretty soon. But let's just dive into the context of the ocean briefly. So the video that you see on the left is from the Darwin project from the group of Mick Follows. This is actually a planet scale simulation. But what you're looking in the colors of the where the ocean is a seasonality of organic biomass, which is coming from the plankton that's arising. And so you see these deep plumes of green and blue. You should think of them chlorophyll showing up in the ocean. Very much like forest across the ocean, across the terrestrial land, we have these massive scales of life in the ocean. And one of the perspectives that you have to think about is, if we deeply care about our planet, we have to understand this life in the ocean for two very simple reasons. They could be fairly selfish. First of all, every second breath that you are taking is coming from these plankton in the ocean, literally half the oxygen on this planet. And then the other part is, which when I first learned about it and I started thinking about this problem, was actually shocking to me, which is almost 50% of all human-generated CO2 currently is being absorbed by life in the ocean. By absorbed, I imply through the biological pump, where this carbon gets dissolved in the ocean, that then gets taken up by organic matter, especially plankton, converted into biological matter, but then sequestered to the deep sea. So literally, every atom of CO2 that our cars are emitting half of those are being sequestered by the most prominent biological carbon sequestration technology that we have on the planet, which is our ocean. And the challenge is we don't even know how it works. And so if somebody had accidentally switched this biological pump off, which is this cycle of plankton taking on the CO2 from the atmosphere, turning it into life, pumping it back, the numbers for the parts per million by estimates are such that our ppm for carbon, CO2, would suddenly rise to 600 parts per million from 400. And that's a little bit scary. But again, one of the perspectives of this is sometimes when we think about the ocean, we often think of it as barren of life. And this is one example of an extreme scenario. This is a dive across the coast of Papua New Guinea in the night, and you just see the abundance of life. And of course, these are more macroscopic creatures. What you're not seeing in there are the microscopic life forms. And we have thought about this from a perspective of genomic discoveries. Hundreds of millions of genes have been discovered that are associated to marine life alone. Half of them have absolutely no mappings to anything that we know from a terrestrial context. So there is an immense amount of biology still to be discovered. But one of the perspectives is, how do you study this biology when we don't have the kinds of tools to really be able to do cellular scale details of these organisms that we care about in the context of the ocean? And another way of thinking about, going back to the paintings that I showed from Haeckel or the Dreyers rhinoceros, here is an example of a few coccolithophores that I'm showing, which are one of the primary sequestration mechanisms for CO2. And what I find remarkable is just also the aesthetic beauty and the remarkable sets of shapes and forms these organisms take, always at least leave all of us wondering to what might be going on. How do I prescribe function to much of what is unknown in the shape and form in this context? And for some of you on the context of sequestration side, the living proof of that is the mountains of Dover, which if any of you have visited UK, you see these little mountains made out of single cells that have all sedimented to the bottom of the ocean. This process is happening at this very moment. But one of the perspectives that we don't have and understand is, how does life decide in the ocean where it wants to be? How does it make decisions of when to go down, when to go up? Or does it actually, microscopic life, has any context of up versus down? So this is what we will try to tease apart today. So let's do a quick quiz. And I think since this is not an interactive platform, I'm just going to give you the answer. But very simply put, the ocean, when you start thinking about, if some of you dive, you know that as you go deeper into the ocean, you start losing sets of colors. It starts becoming more and more blue. And then, of course, after 1,000 meters to 1,500 meters, it starts becoming absolutely black. So there is a euphotic zone, which is between 100 to 150 meters. Then there is a mesopelagic zone from 100 to 1,000 meter. And then after that is the abysmal ocean. There are places on average, the ocean is around 4 kilometers, places where it can be as deep as 11. And a simple question is, if you were to drop a single cell, say a diatom, how long would it take for it to reach, say, the average depth of 4 kilometers? So the question here is very simple in terms of Stokes drag associated with a small cell. You have gravity, you have cytoplasmic density. Cytoplasmic density of most cells can be between 5 to 10% of seawater, roughly. And a simple quiz that I was going to ask you was, if you were to drop a penny from the ship versus if you drop a diatom, what would be the difference in time of how long would it take for it to reach the bottom of the ocean? And actually, the answer is quite surprising. This is just the culmination of Stokes drag. A penny might take a couple hours, while a diatom might take a couple years. So it starts to tell you how slow this biological pump is, and how is it possible that this sequestration actually still happens at such a massive scale. So what we're going to do is first dive into some of the work that has been done in understanding how life positions itself in the ocean, and then ask questions of what is fundamentally not known about this mechanism. So this mechanism is called diel migration. This is the largest biomass migration on the planet. So if you think about the Serengetis, and if you think about all the animals crossing the savannas, that's extremely small in terms of the biomass displacement that happens in the ocean every single day. And the discovery of this was just incredibly beautiful. It started in the World War II when sonar was becoming much more prominent by the Navy. And many of the Navy generals reported that in the middle of the ocean, they started seeing that the seafloor in the night was rising. This was the call that was sent to the head office. And of course, the head officers thought that maybe there was too much drinking or what was going on. They didn't believe it, except then after day after day after day, it was observed using these sonars that literally the effective reflection layer for sonar in the day would fall down and in the night would come up. And so effectively, it took 20 to 30 years to figure out that this was actually a biological matter that was moving up and down, and it was so dense that it would give a very clear diel migration signal, with organisms traveling all the way to almost a kilometer every single day. So if you do the math for this, this would be implying that you and I would run a marathon every day in the morning and every day in the evening, right before breakfast and right before dinner. So we're talking about almost cells, hundreds of microns of cells traveling between 100 meters to a kilometer. And that should give you the context of, if some of you think about cellular metabolism, if some of you think about just ecological length scales, in that length scales, we have a massive transition in temperatures. So if I go back here, I've plotted here what I call thermoclines and pycnoclines. So a massive temperature change from when you are up where you're being soaked by the sun, a massive salinity change. It's changed in pressure every 10 meters. Many of you who dive know that every 10 meters, there's almost 1 atmosphere of change in pressure. So there are many physical parameters that are starting to change for organisms. And one of the questions that we've been starting to ask is, how does life in the ocean, first of all, whether life in the ocean knows where it is from a context of depth, and how does it, given all these sensory inputs, make decisions about where to be? And the big question is, how would you study this, since you're talking about the scale difference where the organism can be 100 micron, but it's traveling a kilometer? So how do I observe distance traveled at the highest possible resolution, but with things that are traveling almost a kilometer? And much of what I'll talk about today sort of depends on organisms that are roughly around 1 millimeter or smaller. Because above that, you can start thinking about the behavioral landscape, for example, of what a whale can do, where organisms can completely overcome the effect of gravity and they can swim at will, while sets of organisms where the behavior is deeply coupled with the physical parameters, which lies right in the middle. And then all the way, what we will not talk about, although it's relevant in this context as well, is, for example, a viral particle or a bacteria, because in there, the Brownian dynamics itself and the orientational disorder that arises is short time scale, such that their dispersion will be dependent much more on their orientational disorder. So we are in the regime between roughly around 1 micron to 1 millimeter, which is majority of the plankton community from a biomass perspective. So this is what has been done in the past from oceanography perspective. Oceanography to me is much more like macroeconomics. We try to measure from a population density perspective what populations are doing. So you could either do genomics, where you would go and collect billions and billions of cells, you would grind them up, and you would give some average statistics of what those cells were doing. You could give with these CTD samples with fluorescence measured as a function of depth. You could measure a layer of fluorescence, which would tell you where the abundance of the organisms were. You could create these things that are called marine snow traps, which essentially trap sinking biological matter. And again, it's a trap that's left for a month. You collect a lot of material that essentially either grows or falls on it, and then you make some statistics to say how much carbon flux that was coming through these. And one of the contexts of these sets of methods is that they're all population-based, they're much more statistics-oriented, and then they don't give you the capture of what's actually happening at a cellular resolution. And I think much of the carbon flux models currently are built on these measurements, and this leads to this challenge, which is, as many of you have realized, the environmental and the ecological challenges that our planets face. This is a paper, I think, from 2017, 2018, and I love the title of the paper because the authors are being very honest. And it says, assessing the apparent imbalance between geochemical and biochemical indicators of meso and bathypelagic biological activity, or what the dash is wrong with the present calculations of carbon budgets. And this is a very blunt, this is a very serious paper, but it's quite blunt in its approach that many of our measurement technologies have not caught on with the computational capacity and capabilities that we have in modeling. But if you don't feed it the right set of data, we will be in a problematic situation in predicting what actually happens in the ocean, especially to the carbon cycle. So that's all I wanted to give you as a prelude. I think I went a little too long in the prelude, but I wanted you to understand and estimate the scale of this problem. Now I'll tell you a little bit about what we have been doing. And I know many of you are cell biologists and just deeply care about the biodiversity of life. I think for many of those contexts, there is just beautiful cell biology to be discovered in the ocean as well. And I'll transition to tell a little bit about some of the motivation combined from the ecological context, but the other aspects combined from the sets of questions we're interested in. And then the big question we're interested in, how does a cell know where it is? Does a cell know up versus down? How does gravity actually get detected in a cell if it does? And that's an open question. How does pressure, a really fundamental parameter of the ocean where an atmosphere is changing, so if you're doing 100 meters per day, that's 10 atmosphere of fluctuation. How does biochemistry change with that pressure? There's a lot of class of questions we are starting to now ask with this tool. And then I would be delighted to talk to you all offline in terms of many of those questions. Today, I'll just get the chance to share with you the sets of tools we've built and some of the tantalizing discoveries that we've made so far. So let's dive in. I think I want you to keep in mind that when you think about the ocean, don't think of it as a homogeneous environment. Think of it as a vertical stratified environment with light, pressure, nutrients, temperature, and essentially all other aspects of physiological parameters to be extremely stratified along the vertical axis. And so you have to ask the question that when a cell goes through its cycles, it's actually seeing a very wide range of biological physical parameters and phenomena. So often enough for the last 200 years or more, many of you, as Martin already mentioned, might know I love microscopes. And, seeing through a microscope is just one moment when suddenly your world opens up. But one of the challenges that we realize is for the last 200 years, we've been studying life under a cover slip. There is a set of a challenge that we often think about life in the XY plane, and we wanted to observe life in the YZ or the XZ plane. And so we started asking, what can we do to microscopes to allow us to study this vertical context? And much of this talk is going to be about flipping your microscopes 90 degrees to account for the context of gravity. But then I'll show you a trick today that allows literally any microscope in the world currently to be reconfigured to allow us to study these vertical migrations. So the gravity axis, it's important to understand, is now perpendicular to the optical axis, which is now in the horizontal plane here. So how do we study vertical tracking? The classic vertical tracking things that have been done so far is you take a cuvette that's long enough, and then you observe, and you can put your microscope on a tracking stage, and you move with the organism. The longest tracks that had ever been made so far for this type of a scenario was between a couple millimeters. And as I said already, that we have anecdotal data from organisms that they can travel almost a kilometer. So you can see almost multiple orders of magnitude of a difference. What we would like to actually do is to watch a single cell, say that's 10 microns or 100 micron, travel 100 meter in 24 hours, and actually be able to study its biology live with the greatest detail that we have developed so far. So you see this gap in technology, and this is what we started realizing that led to the invention of what I'll tell you, which is the gravity machine. The idea is very simple. Deepak and I, one of my graduate students, were scratching our head in the office, and one day we sort of realized, what if you took the two ends of a cuvette and connected them together to form a closed loop? So if I was physically with you, I would have pulled out a hula loop, for example. You can do this experiment at home. And it took us a long while to actually realize the mathematics behind this is quite non-trivial, because you are holding a fluid-filled loop. And the idea was, we call this a hydrodynamic treadmill, which is if an organism swims upwards, we move the wheel down. And if the organism swims downward, we move the wheel clockwise and counterclockwise. So in the frame of reference of the lab, the organism doesn't go anywhere, but in its own frame of reference, it's actually climbing. And then we map the rotational degree of freedom of this simple loop essentially to a virtual depth. So I often explain this work by saying what we have created is the Oculus Rift for single cells. And you will notice the caveat here is that, oh, what if the organism starts swimming in the XY plane? And that's one of the limitations. But the discovery that we have made is, you will be shocked and surprised with this, that ironically, organisms from the ocean don't do that. And they align their motility. And so if you were to plot their diffusivity across the XY plane versus the Z plane, it's highly anisotropic, and they are detecting gravity and actually locked in the vertical axis. So I'll show you data to prove something like that, but this is the simplest thing that you should think about. Now the challenge is that you have fluid-filled tube, so as you rotate, the momentum imparted from the boundary has to eventually reach the surface. And of course, there's viscous dissipation, there is inertia in this fluid. So how do we account for the fact that this fluid might slosh around and hence completely ruin the tracking? So we went back to essentially, so this is the idea. It's a hamster wheel, but for single cells. And it's done through control algorithms where we have a camera watching the organism. This is the actual construction, and I'll dive into the actual instrument in a little bit. So much of the controller for XY, we are measuring both the depth of the organism. We're doing live tracking, and this is the actual instrument built in, and you see this glass wheel. This glass wheel is on a rotational degree of freedom that can rotate, and that gives us the access to be able to lock in an organism and travel across. But again, as I said before, because there is no boundaries in the vertical axis, we can essentially do this for infinite distances in the frame of reference of the organism, in the vertical axis. So the simplest way of understanding this is to think about the sets of conditions that would be set for you to be able to do this. And you can break down the context of the time scales that are associated with this. First of all, there is a time scale of delay of the fluid, and there is a time scale in the delay of the stage, which comes from the control parameters. And of course, there is a time scale of the behavior of the organism. So how do we know which sets of organisms we can track? So here's a phase space. You see from green to red, the green region are the sets of parameters below the red line, which we can track successfully. And above the red line are unsuccessful locations because the organism was just too fast for the fluid inertia. Otherwise, if we move the stage too fast, it would impart inertia in the system, which is not what we want. But to our surprise, almost, we have now tracked around 75 or so species, and there's only a single species that kind of fits and goes above that red line. And this just happens to be a coincidence that nature has been kind to us that allows us to be able to track almost all the organisms that we have studied so far. Similarly, there is another condition that we apply that, as you can all imagine, because from the boundary, as you move the boundary, there will be a shear term associated with this, and that will apply a small shear to the organism. And we account for, depending on the speed that we will rotate the wheel, for this shear to be below almost 2 orders of magnitude of the shear that's essentially generated by the gravity to make these organisms stable. So there is a stable regime and an unstable regime, and primarily for all the data that I'll show you today, we're in that stable regime. So we do not perturb the orientation of the organism because we believe that the organisms detect the orientation of gravity and align themselves with gravity. It's very important not to perturb, but we do have a handle. If we would like, we can actually flip the organism upside down as perturbation, which I don't know if I have time to show today, but we can talk about offline. Okay, so let's just dive in and talk about, let me skip much of this because I want to come to the data. So here is the sets of parameters. Depending on the geometry of the wheel, we can decide what sets of parameters will account for this optimal rate of tracking, and when we will have a viscous time scale that's capped such that we can keep the organism under our watch. So you can write down these 2 conditions, and as long as these 2 conditions are met, we are in a regime that is trackable. And of course, all of this happens to be in low Reynolds number regime, even for organisms that are, some of them are 500 or so microns. So let me just skip this. This I already walked through this, but this is the display of how the system works. This is a time lapse that has been sped up. So you saw the wheel was turning. Of course, the wheel is not turning this fast, but all the data that I'll show you today is collected in the field. So the instrument that we've built is portable, and we take it both to marine stations, and actually we just got back in December before the shutdown from an ocean vessel. So I'll show you some data from the middle of the ocean, which is the first time any data has been collected at a cellular scale from organisms that were just plucked out from the ocean and undergoing vertical migration. So I'm hoping I'll get a chance. So now I'm going to talk a little bit fast because I'm realizing with this virtual platform, I'm not keeping pace, and I want to show you much of the data and the biochemical puzzles. So I apologize in advance if I'm going to go a little bit faster. So let me skip all of this. There is a lot that went into building the controllers, but one of the joyful aspect of it is every single time we have put an organism in the system, we have observed something completely new, which is, to me, the essence of science is not imposing our own biases to what we will watch, but to truly just sit and watch. And, this is one of the moments captured in Puerto Rico when Deepak and I discovered vertical migrations in diatoms. And I'm going to show you that data pretty soon. Okay, so let's dive into what do you see. First of all, if you put beads, and I know some of you care about applied math and fluid dynamics, here's a classic puzzle. It's called the kiss and tumble. So if you just drop dead beads, what you would see is you would start seeing hydrodynamic interactions. So the movies are slowed down here somehow, but if you watch, what you're seeing are these beads traveling. They're falling, but you don't see them falling because you're traveling with them, but you're starting to see them interact. So you can think of this much more as a multi-body interaction problem, while the beads are generating hydrodynamic fields that are seen by each other, and hence they can interact. To the very right, what you're seeing is how, go ahead. Was there a question?
The questions come at the end, so
okay.
There's some interference. So yeah.
Okay. Can you see the videos, or they're kind of bumpy?
They're kind of bumpy, but we can see them. Yeah.
Okay. Yeah, maybe I'll change something on my computer to just see whether it's coming from my computer or otherwise. So let me just stop that for 1 second and see whether, yeah, it's coming from my computer. So what you're watching to the very right here is every time you put sugar in your coffee, that sugar piece dissolves. And what you watch was this dissolving trail, but now we have the capacity to track that vertical migration, and hence we were able to actually see that the trail. And there's lots of questions we're asking on self-similar shapes when passive objects dissolve. But one thing that we're going to do is dive into living things, because living things follow criteria that are dramatically different. So let me just skip this for a second. But this is the point of what truly ends up happening when you throw a piece of sugar in your coffee, and what happens to the fate of that sugar. You're seeing these two steady vortices that are attached to the bottom, and you're starting to see the shape of this sugar change. It's a dissolution problem as a function of sedimentation. And one of the questions we are trying to ask is, what is the shape evolution of a sphere as it falls? And ironically, such a simple problem has actually never been solved. So there are lots of mathematical problems that we are interested in from a context of sedimentation. But we're going to dive into data from real organisms. And so one of the things that you're seeing in this video here, and again, it's a little bit shaky. I'm going to try to see if I can close other programs on my machine to see if my machine could go faster. Just give me 2 seconds here. If I closed everything, my hope is that it will not close the Zoom link. Okay, so we will try again to see whether these videos become a little bit better. Yeah, maybe not. Okay, let's keep, actually, maybe. So what you're watching is literally, so all the way from acorn larvae to brittle stars to the right is actually a diatom, and you're watching that diatom fall. And I'm just going to focus on that data for a little bit in a few minutes. But one of the contexts is we study all the way from embryos. So I know many of you are interested in developmental biology. We've been asking a question, as a single cell develops, how does it decide where it wants to be in the ocean? So we've collected data based on, one of the tantalizing discoveries that we have made is how the gastrulation axis and the axis of an embryo is connected to gravity, all the way to single cells, that when cell division happens, how does that get coupled to the behavior of where the cell wants to be when it's undergoing, say, its cell cycle for division. So let me just show you some of the data. So the first set of data is around marine snow. And one of the big questions that we are interested in asking is, how does dead organisms, because much of the carbon sequestration that I talked about happens in dead organisms, how do they find each other? How do they form these marine snow particles that become heavier and fall? And how does the microbial life essentially chew that up? This is a very large question of what is called a phycosphere, because if the microbial life chews this dead organisms faster, then you don't get any carbon sequestration. And what you're looking on the right, it's to the very right of your screen, is the first data set that we collected on marine snow. And what we have is a single cell resolution on this dead aggregate of particles, of which are diatoms, but the length scale in this data set is 10 meters. So we have watched this organism fall and change as a function for 10 meters. And these type of data sets are really hard to actually show, primarily because of the multi-scale problem. So now you are watching that single cell and this marine debris falling while, and of course it doesn't look as it's falling, but because we are in the frame of reference, while the actual length scale that we are tracking it for is 10 meters. So this is what matters in terms of thinking about microscopic and macroscopic data combined together. And one of the beautiful things that we discovered with this data set is the porosity of this marine snow, because we can measure the flow through this porous structure. And based on that, we can calculate porosity that actually tells us the sinking speed. So classically, what has been done for marine snow is you apply the spherical cow problem to say, I roughly know the mass of this object. I know that the size, and from that, I'll calculate how quickly it falls. And it actually turns out that's a huge overestimate, and it overestimates carbon sequestration because it makes these marine particles appear to fall faster than they actually do. So here's one example that by just watching, you can immediately correct estimates that we use in global models. So I'm going to switch to something more alive, which is now the context of many marine organisms. And what you're watching are tracks of many different species. So this is 7 to 10 species. And we notice something remarkable, which is these organisms are free to move. First of all, they have behavioral complexity of their own, but ironically, they are actually coupled and aligned to the gravity axis, even for organisms that are actually swimmers and have the capacity to swim in any direction. So the identity of these organisms range all the way from these embryos, which classically, if you were to put them in a beaker, will swim in random directions. When you really put them in these vertical columns, they will align to gravity and they will show these extreme gravitactic behavior, all the way to single cells and dinoflagellates that show these extreme vertical swimming behaviors where they align their orientation vector. And one of the things that we can do with this type of data set is really compare and analyze them and try to ask based on the hydrodynamics of what it generates, because we can also capture the flow fields around them to understand what is their propensity to align with gravity. And I'm going to skip much of this, but this is a very busy plot just to show you that almost everything that we have looked at in the tree of life that falls in this length scale that has evolved in the ocean has this behavior of aligning to gravitaxis. So gravitaxis is a fundamental phenomena that is extremely ubiquitous for life, aquatic life primarily, but also life in the ocean, primarily because the ocean landscape is such that you really need to know where you are and which way is up versus down, because there is an environmental and an ecological patterning. So the ecological pressure along the vertical axis is so strong that we see abundance of behavior that aligns to the gravity axis. And of course, all of you are wondering how that actually happens on a molecular scale, and that's a mechanism that we're working on that would be a story for another time. But I want to just share this data set first of all. And of course, there is a remarkable amount of complexity in these data sets. You can zoom in and look at these individual trajectories. And one of the context of this is that we can compare all of these data sets on a single sets of metrics. These are behavioral metrics that we are starting to develop that allow us to understand the strength of this gravitaxis. And not all organisms demonstrate an equivalent propensity of gravitaxis. So in another way to say this is they're not slaves to the gravity axis. This is not a passive sinking, because of course much of this behavior is equivalent in rising as well, but it is active alignment to gravity, and that degree of alignment varies between different organisms. And the way we can do that is we have perturbation experiments where we will switch the gravity axis by rocking the entire apparatus just a little bit and watch the perturbation and how long it takes for the organism to detect that orientation and align with it. So you should be thinking of this as active alignment to gravity. And this is diel migration. So what you're watching, for example, on the left is the same organism swimming down in the day, and that same organism now swimming up at night. So as I had said before, the vertical migration, this is the largest migration. Now this is a cellular single cell view of those sets of migrations. completely different behavior as a function of circadian rhythms and the light patterns. And one of the things that I'm just going to do is I'm rapidly showing these sets of behaviors because I want to get to this diatom video soon. So I think I already showed the Pteria miniata video. So let me just actually dive into the diatom data. So here is the puzzle that we are working on. We have some clues, but not all of the aspects of these puzzles are solved. So I want to share with you something that's an open paradox right now. So this is the data that we collected in Puerto Rico out at sea. This is for a centric diatom. And what you will watch is, so this is, 100 micron organisms, and as you would have thought that it should sink, but it doesn't. Now, diatoms are not known to have any flagella or cilia or anything. So how on earth is this organism, and the transitions that you're seeing are the organism is falling, and then suddenly, within hundreds of milliseconds, it can literally freeze itself in a water column. So the only way to be thinking about this is that the mean cell density has changed dramatically between 10 to 20% within hundreds of milliseconds. And of course, the organism, this is a behavioral response. So you remember when I had said about, oh, how long does it take for a diatom to fall from the top to the bottom? Actually, the answer of that is unknown because diatom decides whether it wants to fall or not. And if you think about this, it should lead to an immeasurable number of puzzles in the sense that how can the cytoplasmic density fluctuate at these large scales? And there are several sets of hypotheses that we're working on. We do these sets of assays in the presence of drugs and without drugs. And one of the big theories that we are working on is fast ion channels, the capacity for ion channels to open and close and regulate this density in short periods of time. So that's all I'll say about this diatom puzzle. But this is what I meant by just observing and watching organisms in their ecological context. We have studied diatoms for the last 100, 200 years. And one of the things that is not apparent immediately is that with these long-term tracks, we have the capacity to see behavior. And ironically, almost a couple of months ago, there is a class of papers that have come out that tried to patch clamp diatoms, and they seem to show now on a stationary XY microscope, and they seem to show spiking behavior, very fast action potentials measured from a diatom. And again, I think one thing that we've been trying to do is connect those observations with these density fluctuations to be able to see whether truly these sets of action potentials might be responsible for these fast fluctuations. We can do a lot of statistics on this because we can collect this type of data for 24 to 48 hours continuously on a single diatom. We can watch this same diatom divide and go through its cell cycle, trying to connect where it wants to be in the context of cell cycle. And this is the importance. We can do all of this with classical techniques such as fluorescence, epifluorescence microscopy. So what you're watching is the same diatom now, but with autofluorescence from the chlorophyll of that sinking diatom. And this was a lot of work to be able to now get our highest resolution microscope be coupled with our controllers to be able to do this multi-scale data. So since I'm almost out of time, I wanted to show some cell division videos, but what I'm going to show you is show a video or two of what happens when we take this instrument out at sea. So in December, right before Christmas, we took the instrument to Kilomoana. So this is, how do you really do cell biology in the ocean? It was my first trip on an expedition, and I'm very thankful to the Kilomoana team to allow us to give some space to come. It was extremely rough seas, and I'll show you that. But this is the first sets of videos that we took of gravity machine operating out at sea, where we collect organisms at a known depth. We bring them rapidly up, and then we put them in the gravity machine. The time duration it takes from how long we collect the time to the time, and I think one of the ironic things about this moment was the storm was coming, and storm is here. Once we had proposed this experiment, nobody believed that we could actually do this delicate cell biology and this tracking microscopy at sea, because of course, any of you who've been at sea, the biggest challenge is that the boat is rocking. And we figured out a way to decouple the rocking of the boat. And that's the thinking where we collected the cells from. So the cells came out from this line. We get them out, and with 15 minutes or so, we get them back in the machine. And because we know which height we collected them from, we can virtually assign the same pressure and light conditions in the wheel to allow us to observe the cell's journey beyond that point. And one of the contexts of this is all data now collected live at sea. And one of the things about this was, for us, for the first time, we started seeing for many organisms in the lab, in cultures, we would always observe sinking behavior for these passive organisms, but not the rising behavior. But the first data set that we took with diatoms, we immediately saw rising behaviors, which again speaks for the fact that when you have cells that are collected out in the ocean, you have physiological recordings from them where the behavior is mapped. And I think one of the things that I'm going to leave you with was all of this data was collected. At some point of time, we had to shut down our experiments. Turned south, and this is what it looked like at some point of time, early morning. And we were out there for all and cut short by primarily because of weather conditions. But I think it was a great lesson for us to really prove to ourselves that, yes, we can bring these types of instruments. This is another example of what happens. I'm just showing you some of our failures as well. I didn't mention this, but we have built the machine that allows us to regulate pressure inside this chamber. So we can, at this point, do around 20 atmosphere. And of course, one time while experimenting, we went much higher, and of course the wheel exploded. But these types of things happen, and we are starting to design these wheels to have the capacity. Of course, temperature we can already change, light we can already change, pressure we have the capacity to change. We're working on ways of also changing salinity, because salinity is a big important parameter from a density perspective, because salinity gives rise to density fluctuations in the ocean. So there's a lot of improvements that are coming on in this technology. And I'm going to end here by just stating that, to me, the joy of cell biology lies in connecting the behavioral context of live organisms. And many of us who study live cell biology and live organismic biology know that watching a living organism is just such a joy. Connecting that with cellular physiology, but then also making sure that the environment is not forgotten. Because then how do you really bring the ocean to the tables in our labs is really been the big goal here. And at this point, I will end by just mentioning that my hope is that we can make better rhinoceros next time, because we have seen them in what they truly do out in the ocean. So I'm going to end by thanking all the people in my lab. There's a big group of us focused on marine context. We spend a lot of time at marine stations. And one of the goals is to plan a trip to a few marine stations in Europe. So the next time we are close to Europe, maybe I'll get a chance to actually visit all of you in person.